So before we got cut off, we were at this slide here. So let's take a look here where we're at. I'd pulled up a couple of points and we were talking about the fact that you have the first tRNA come in and bind into the P site, and it gets locked in by the larger subunit. Now, what's important to note is there is a difference between prokaryotes and eukaryotes, and that first amino acid in prokaryotes has actually got a formal group added to it. So that's why it says FMET here, formal methionine. And there you can see the final point is talking about the larger subunit attaching, and it locks that P site with the tRNA that contains the methionine. So it locks it all in place, and now you say that the reading frame is in place. So now the ribosome is going to be able to move along, and new tRNAs are going to be able to come in. So there you can see the tRNA being locked in at the P site, and you can see the codon and anticodon complementary nature. Okay, elongation. If you've watched that animation, you would have seen this happening. So I'll pull up the points here. Okay, so now that you have the P, once the P site is occupied, the A site is open. So, and it's going to have its own codon, and that means the next tRNA that has an amino acid attached to it is able now to come in and bind, and it is complementary to the second codon, and it occupies the A site. So right now you have the P site and the A site occupied with tRNA. And what happens is when those two sites are filled, a peptide bond is going to form between the two amino acids. And that's done by the enzyme Peptidyl Transferase. Okay, you can see that happening at this stage here. Now the first tRNA is free, and it is free because it has been shifted to the E site and is going to be ejected off. And the ribosome is moving down one codon, and that's really what releases this one from the E site. So you can, this one here, there's the transfer to create the peptide bond. Now it has nothing on it, and the ribosome is going to shift down one spot. So what was here gets shifted to the E site and gets ejected off. And what was at the A site has now moved to the P site, and that opens up the A site for the next tRNA molecule to come in with its amino acid that is complementary to the next codon. And this process continues over and over again as the ribosome shifts down the mRNA, exposing a new codon. A new tRNA comes in with the amino acid, and you have a polypeptide created. That is until you reach a stop codon. So the end of the mRNA is a stop codon. Remember, there's 3 different stop codons. So when the ribosome reaches that, and that is now exposed at the A site, there's no tRNA that matches up with it. So no tRNA comes in. Instead, what happens is a release factor protein comes in. And what that's going to do is it's going to cut the polypeptide chain from that last tRNA. And now the polypeptide is released. And the polypeptide can now form its 3-dimensional shape, undergoing secondary and tertiary folding. And the ribosomal subunit separate, and everything is dismantled. So you can see that happening here. It has reached the stop codon. There's the tRNA, the last one that has a really long polypeptide chain. So instead of a tRNA coming in, a protein does, called a release factor protein. And with the addition of some energy, everything dissociates and the polypeptide is freed from the tRNA, and it can now undergo its folding to become a functional protein. This is just an overview of the entire process. So you can see transcription, there's the DNA opening up, there's the mRNA being made in transcription, then there's the modifications to put the cap in the tail and to remove the introns. Now you have the mature mRNA that's going to leave through in the nuclear pore. It's going to get attached to ribosomes, and then this tRNA molecule is bringing in amino acids, and a protein is created. Here's just another slide showing an overview of the whole process of gene expression. And that is the end of this PowerPoint lecture.